So for week one in the lab, we're always looking at lab safety because to be in a micro lab, you have to make sure that you're using very safe techniques. And some of the techniques that we use are called aseptic or sterile techniques. So basically aseptic, sterile. And there's different types of things that you'd be using in the lab, like a pipette, inoculating needle or loop, cotton swabs, etc. Now we also have to keep in mind that there's dust in the air, particles in the air that we don't want to get in our samples. So before you start, you should always be wiping down your counters with disinfectants. And if you do spill something, we do have disinfectants in the lab that you would use to clean that up. And you should always be washing your hands. Hand washing is extremely important throughout the whole semester whenever you're in a lab. You always make sure that you're carrying tubes in a test tube rack. And again, make sure that your Petri dishes are media side up when you are incubating them to reduce condensation. Now, some basic things that you want to do. First of all, always work by a flame. It's extremely important to work by a flame when you're working with microbes. You should always be heat flaming your inoculation tools, whether it's a loop, whether it's a probe, etc. Make sure they get red hot and that they cool before you actually put them into the culture that you're trying to get the bacteria from. So once you've done that and you inoculate your media, what are the types of media that you could actually use to study microbes? Well, the first one would be an agar plate. And on the plate, you could look for different colony characteristics. Now, a colony is a group of cells that arise from a single cell. Again, a colony is a group of cells that arise from a single cell. Good to know for your test. So, what would you be looking for then on an agar plate? Well, shape could be circular or regular, etc. They come in different colors. Look at the margin, like around the edges in that, guys. Also, elevation, if you hold the plate sideways, is it flat or raised? Texture, could be dry, mucoid, or moist. Now on broths, those are liquids, so you would look to see if it's cloudy or if you have chunks in there. Sometimes you get like a little film or a membrane on top, and sometimes things sink to the bottom. Now slants, also, we have Auger Slants, guys, that you could look to see, is it aligned? Does it look crusty? Is it like spreading out? Again, you could see pigments in to see whether or not it's transparent or translucent. So let's just take a look then. These are similar properties that I mentioned for plates that you want to familiarize yourself with. Now, if you take a look here, this describes some of the whole colony characteristics like punctiform, real small, circular, rhizoid. Look at the edge. If it's undulated, it looks like that, etc. Now, if you take a look here, for instance, at these two colonies, guys, what you can see is this one is circular. It looks smooth around the edge. This one over here is filamentous. You can see these filaments coming out all over. And in the broth situations here, this is uninoculated, beautifully clear. Here, though, it's a little bit cloudy or what we call turbid as well. Now on Auger Slants, as we see here, here's some drawings of the different growth patterns. This is what the actual auger tubes would look like. You can see that we have this filiform growth on this one and what we call effuse or spreading growth on that tube. Now, for your first post in the lab, I would like you guys to post something on hand washing. So you can go to the CDC website, you can just do a Google search, but find information on why hand washing is important. And please note this gentleman right here, Ignaz Semmelweis. So Ignaz Semmelweis is the first person that suggested that people wash their hands when going from one patient to another. Now, hand washing, what it does is it actually removes microbes from your skin. Now, the idea is you wash long enough, let's say 20 to 30 seconds. Make sure you get a good lather, warm water, warm to hot water. Rinse it off, life is good. However, we do have what are known as normal microbiota. So please know, guys, for your test and quizzes and such, that there's three main groups that can be found on your skin. They're called diphtheroids, staphylococci, yeast, and fungi. So they're normal. These are things that should always be there on your skin. So even if you do a hand washing experiment properly, and please watch the video that I uploaded for you on how to do that, you still might find these normal microbiota because those are things that should be there all the time. So since we're not doing it in the lab, you don't have to worry about this protocol, but this is normally what we would do in a face-to-face lab right here. Now, I also wanted to just go over quickly the first couple of pages in the lab manual. Now, when we talk about lab safety, a few things that you should know, and this is page 5 and 6. In the lab, always wear a lab coat, always be washing your hands, tie back your hair. You should never have open-toed shoes in there. No food or drink, no smoking, etc. Be very careful with the chemicals that you're in there, and don't take anything out of the lab. Again, before you start and when you're done, always wash your hands. That's very important. Other things that you guys want to do, make sure that your Bunsen burner would be off. Again, don't come to the lab if you're really sick because you don't want to be working with these organisms. They are live. So pregnant women, usually there's no problem in the semester, but it's always good to let your instructor know. I would show you where the first aid kit is in the lab. Again, please look at the video on lab safety. It shows where all of that is in the micro lab. Now again, something to keep in mind, before and after lab, always wipe your bench down, should that show up on a quiz. Always wipe your bench down before and after class. Clean any spills right away. Don't worry about it. Telling your instructor, mistakes happen, guys. You got to make sure you do it. You should always have your, notebook with you and those kinds of things as well. Now, again, important things that you should be aware of because these will show up on your quizzes and tests, guys. How do you dispose of things? Well, anything sharp like a microscope slide needle, sharps container. We have reusable tubes. They will be autoclaved and recycled, so they go on the test tube rack at the front of the room. Any other types of things, like broken glass, etc., again, that goes in a biohazardous sharps. Now, also, throughout the semester, we're using auger plates. So you would tape those up, put in a biohazard bag, and that biohazard bag's always on the autoclave cart. Now, please know, guys, that there are 4 biosafety levels, 1, 2, 3, and 4. And please note that we use bacteria that are considered BSL-1 and 2. We use BSL-1 and 2. Why do we use that? Well, they have a very low risk of infection if you use the proper safety precautions. Okay? And throughout the semester, I'll talk to you a little bit more about some other things as well.